All right, without further ado, I will like to invite our next speaker, Dr. Nicole Coufal, who after many trials and tribulations through her journey here, finally made it at 3 a.m. last night. So, valiant effort to get here and meet you all. But she has been, she's a pediatrician. She's an associate professor of pediatrics and neuroscience at University of California, San Diego. And she is a past recipient of the ECDGA research grant. She is very invested and interested in rare pediatric or rare neurological disorders. And in the context of ECD and LCH, it's very critical now. And something we deal with routinely is this phenomena, the term that is used is neurodegeneration. So her lab has been working on that. So we invited her to come here and talk to you all about what she's been trying to do and where the field is moving forward. So please put your hands together to invite Dr. Coufal here to share her thoughts.
All right, thank you so much for having me. I got in at 3:30 this morning, so you'll have to pardon me a little bit. And so I got asked to come talk today a little bit about what funding from ECDGA meant for my lab and for this project and what we're trying to do. And it was really transformational. Wow, that sounds really loud. And so I like to start with this picture because this is the patient that started this journey for me. So I'm not an oncologist. I'm a pediatric critical care physician who does a lot of research in neuroimmunology. But this little baby, this is him kind of after his LCH diagnosis, was in our ICU, and I met his family through their kind of diagnostic odyssey, and as they kind of researched histiocytosis, and it was the first time I had seen it in many years when I was just starting my lab, and his mom kept talking about neurodegeneration and how she was worried about it, and I thought I should read about this. This is what my lab does. And so they were actually the first patients that I enrolled on the journey of this project that I'm going to tell you about today, and really having support from this alliance was really instrumental in getting us off the ground 5 years ago, and a little bit about what we're trying to do now. So I don't need to tell you very much about histiocytosis in general, but the part that interested me was this question of neurodegeneration, which in a lot of the different reviews is a little bit this black box of why this happens and how this happens, and is generally kind of often more treatment resistant than some of the peripheral disease. But there's this idea that the precursor cells with the MAP kinase mutations take on a number of different myeloid phenotypes that contribute to neurodegeneration. And really the question that I was interested in as somebody with a PhD in neuroscience was this phenomenon of neurodegeneration, which is often more treatment resistant, is often progressive, that we don't understand that well, and like what are the barriers to understanding that and to identifying new treatments? And why are the treatments less effective for neurodegeneration than they are for peripheral manifestations of disease? And so these were what I thought were the unanswered questions when I started this 5-6 years ago. So why do only some patients get neurodegeneration? Why are the types of brain lesions patients get so variable? And how does that heterogeneity impact the treatment response? And my background is in stem cell biology. Why do I think that's an important model to have? Like, this was very reliant on patients, including the patients that I saw and got samples from, as well as funding-wise. So I think brain biopsies are really rare. So there's not a lot of lesion samples, not a lot of kind of opportunities to study some of those. There's a number of mouse models that have been generated of the MAP kinase mutations to try to understand why this works. But the immunity of mice and humans is incredibly different. Like, mice can survive anything, right? Like apocalypse. Their immune systems work very differently from ours, and for things like bacterial pathogens, it takes 1,000-fold higher stimulus for them to get a response. So a lot of times their immune systems don't match what humans have. And most of the mouse models of the different histiocytic disorders don't get any neurodegeneration at all, and none of them get any of the heterogeneity that patients get. So trying to understand that. And so I thought, well, let's try and use some of these patients, especially that first patient that I saw, and try to ask some questions around, can we make a stem cell model, and can we try to understand this disease better? So I thought we needed a human model, and that's what we spent the last 5 or 6 years doing. And we do that with something called induced pluripotent stem cells. And so induced pluripotent stem cells, if you've never heard of them, it's basically taking a sample from a patient. That can be a skin sample, that can be a blood sample, some kind of differentiated cell type, and using some transcription factors or core kind of things in a dish to push those cells back to a more primitive state in the dish that can then make all the different cell types of the body. So what you're trying to do in a dish is be able to make the cell types of the brain that you don't get in from patients very often. Nobody ever wants to give me brain samples. So, and be able to then ask, how do those respond to treatments? How do those respond to drugs? Can we find new therapies and new ways of understanding this? And so that's exactly what we did. And this actually just got published in the last month in Blood. I'll have a QR code at the end if you'd like to look at it. But what we did was take blood samples from patients, starting with the BRAF V600E variant, which is being the most common and the one that's most associated with neurodegeneration, and where they have both cells in their blood that carry that BRAF mutation and cells that don't, to try to make both kinds of stem cells from the same patient to be able to ask what happens for the ones that do carry that variant, and how much does the patient's genetic background kind of influence what happens with those cells down the road. So I think each of us has very unique genetic background, and we don't really have a great understanding why some people end up with Rosai-Dorfman, and some people end up with ECD, and some people end up with LCH, and can we try to understand this a little bit better? And nobody had done this before, so this was kind of a journey for us and for my young lab, especially we started this during COVID. But what we wanted to do after we kind of made these stem cells is to be able to make the cells of the brain from them, the myeloid cells of the brain from them, and ask what happens, and can we now ask some questions around what are the specific phenotypes. And I think one of the hard things about using patient biopsies or patient samples is you have this heterogeneity, and then you have all these other infiltrating cells because they are recruiting T cells and other cell types into the lesions. So this is a way to be able to look at just that initiating cell type and be able to ask, how do we get rid of it, right? Like, it's basically a cancer. How do we now target this? And what we really wanted to see was, does this look like the patients? When we do this in a dish, does this look like what comes from our patients? Because if it doesn't, then it's not that useful. And I think some of the things that we found that were really, I think, reassuring that this does work. So in the middle here is one of the biomarkers that's come out of the Langerhans Cell Histiocytosis patients, is that osteopontin is high if you have neurodegeneration, so that happened in the dish. And then we also compared it. This is a heat map on the far end where you can see that the similarity of our brain myeloid cells in a dish was very close to that of a set of histiocytosis patients where they actually had whole brain RNA-seq. And this was a combination of several different histiocytic types of patients. So I think this is actually a pretty good model. We're really pleased with what we're going to try and do with it next. But really, we wanted to see what happens in the brain. And so one of the kind of things my lab has really developed in the last couple of years is a mouse model where you can put human myeloid cells into the brain and ask, what do they do? The brain environment is really important because we know that in many of the histiocytic disorders, what happens in the brain is really different than what happens in the skin or the bone. They don't express the same markers. You have more infiltrative, less single, lesional disease often. And so trying to understand how that works, I think, is really going to be important because we know peripheral disease is generally much more treatable than that of the brain. And so just in a very briefly, I think one of the things that we saw that was really interesting and really made this very unique compared to the mouse models of histiocytic disorders is that mice, when we put in one starting cell type, a homogeneous cell type that carries this, the BRAF V600E variant, is that we saw the whole diversity of things that you see in patient brains, including infiltrative disease, these foci or granulomas of disease. So I think in that sense, it's gonna give us a lot of information about how interactions with the brain environment lead to very diverse kinds of disease and hopefully also kind of treatment pathways. That's ultimately all of this work has been leading up to wanting to ask those questions. And this is from publications on patients, so you can see these same kind of disease foci or granulomas that are seen in histiocyte patient brains is what we're seeing in this mouse model. And then ultimately we asked, do mice get sick like people get sick? And they did, so they had some of the classic kind of motor coordination problems. They don't clasp properly, they can't stay on a rod that's spinning. So it's very basic questions about coordination, but they actually get a lot of the phenotypes that patients get. And without treatment, they have early mortality. So we're really happy with this kind of as a big picture question of how this works. And so I think we were so grateful for the funding to be able to kind of get this project off the ground in the very beginning. There's a lot of other data in here I don't have time to tell you today, but I think this heterogeneity of having both proliferative kind of cancer-like early cells and then more fully differentiated destructive inflammatory cells, being able to isolate those in a dish and try to figure out how drug-resistant or drug-sensitive they are. And they were apoptosis resistant in the kind of senescent state will hopefully give us some ideas what to then try in the mouse in terms of drug therapies or combination drug therapies and help what we really want to do, what I really want, I'm not an oncologist, and I tell people this all the time, is hear what are the things you would like to see from this model in terms of what can be translated to patients, because really like the goal is to be able to do something that's meaningful. So what does the future hold for us? So what we want to try and figure out is first, in a dish, how are these sensitive to BRAF and MEK inhibitors? We know patients have a lot of disease recurrence if they stop therapy at pretty high rates. So can we ask some questions around what the types of populations of cells are that are treatment resistant and what we could maybe add to get rid of those, especially in the brain? And then trying to ask kind of those questions about relapse, transient responses after BRAF or MEK inhibition, and how else should we try to target these? And can we try to generate some data that's clinically meaningful in that setting? And so the ultimate goal is, of course, to develop new therapies and to try and improve patient outcomes. And I have a ton of acknowledgements. I have a really wonderful lab. We do a lot of rare disease work in mitochondrial disease and other areas, but this has been like a passion project. I still see that family of that little boy. He's like 6 years old now. And we worked a lot with Carl Allen, who provided some of the patient samples. So we made a bunch of different cell lines from patients in order to try and ask some of these questions. And I'm so happy to take any questions you might have.